So now after coffee break, I don't need a microphone, but okay. Okay, now after the microphone, after the coffee break, it's my pleasure to start the seed phenotyping session. So for those who know me, they know that seeds for me is like a candy store, so I'm very, very happy. And it's my pleasure to introduce Stéphane Nicolas. And let's go for it. Thank you.
Thanks, I am not a specialist of seed phenotyping. I try to say something that maybe if I have passion. I try to say how we can use it on from plant genetic resources on how it could be useful for breeders. And I changed a little my title as like that, predicting seed composition on agronomic traits in my genetic resources using phenomic selection based on nears. Okay, it's work. Just a few information about maize, but I am certain you know it. Maize genetic diversity was shaped by domestication, expansion, and adaptation to different environments. Maize have been domesticated in Central America, close to Mexico, on spring South on North America, and after have been introduced in Europe, leading to the formation of different genetic groups that are adapted to different agroclimatic conditions. And what is important is its association with a very drastic change in for phenology, but also a lot of traits is well illustrated by flowering time with a shortened time and with photoperiod change from Central America to North. And land races were well adapted to different agroclimatic conditions. And for this reason, we think that there is original and promising alleles for breeding to fast climatic change, but also low input agriculture soils. Genetic diversity of current elite hybrid line was shaped by a transition from land races, open pollinated varieties, to F1 hybrids, following the discovery of heterosis by Schuland East. And here is illustrated by the change on from land races. Some hybrid lines was derived by single seed descent to produce the first F1 hybrids, and after there is reciprocal recurrent selection to conduct to the formation of elite breeding germplasm. But when you trace back to the founder of breeding programs and main breeding programs from different seed companies, you see that as illustrated by Lancaster support derived lines, that it was back to few land races, and we are pretty certain that from land races to first inbreed on to elite germplasm, we lost a lot of diversity, and not too much, but a lot of interesting diversity for traits. On that point, few land races contribute to modern breeding germplasm, and recurrent selection erode progressively genetic diversity. We lost allelic diversity, and for this reason, we think that land races could be one type of source of genetic diversity for pre-breeding and new traits required for low input agriculture and for facing climatic change. And what is important to think is that there is a large number of land races on hybrid line maintained ex situ in gene banks as seeds, and also in situ, but not too much in Europe, to be honest. And for example, in CIMI, there is 26,000 land races that are maintained. In USDA is like 30,000. And in INRAE, just one gene bank in Europe, there is 1,600 land races. And it's the same thing for hybrid lines. And in the world, we think that we have more than 100,000 associations. And it's, as has been said by different speakers before, there is limited use of inbreeding program by farmers. And there is several reasons for that. It's due to lack of characterization, gap in performance, but also for land races and maize land races large within genetic diversity. And we need to low-cost approach like genotyping NIRS for characterizing land races and hybrid line for traits, including seed compositions. And now, how to retrieve at low cost the genetic value of maize landraces is what has been interested for breeders, on farmers, and so on, including seed composition for human use and so on. On first part, we can use predict traits of landraces in gene banks with genomic selections. And to do that, we develop a DNA bulk approach based on 50K array to genotype landraces to obtain allelic frequency, because large within genetic diversity. It allows in the framework of EVA maize project to characterize a large set of landraces for their diversity and organization. And as has been said by Sandra, it has been phenotyped in a large multi-trial networks. And we can combine that by developing some genomic selection approach based to predict traits in landraces. And here is we can use genotype a training population set that has been evaluated in a network to predict and phenotype landraces based on their genotype in GenBank. And we just need to estimate kinship based on genotyping data, and it allows to predict new individuals. And it's very efficient method, providing that you have low-cost method for genotyping. And at this step, we have 1,600 landraces that are currently genotyped with 50K using DNA bulk approach. And it's a database to identify promising source of genetic diversity. And we try by recycling old data in which we have field evaluation of European and American landraces from compared regions in different locations with 2 replicates with genotype with 50K. And we use this approach to do genomic prediction on 1,000 temperate landraces genotype with 50K. And it works pretty well. Here is just an illustration that we can map these flowering times. And fortunately, landrace from South France are late, and in some regions is early, and it works very good. Okay, we are now in seed phenotyping sessions on how to predict seed composition of landraces accession in gene banks. And this is my second part, and I am only 3 parts. We can use the same approach. Near is a low cost. Everybody knows this approach here, I imagine. A non-destructive approach classically used in breeding programs to evaluate seed biochemical composition or digestibility using NIRS on seeds or silage. And this biochemical characterization is based on model trained on a set of seeds samples that are analyzed both with NIRS and with wet lab. And it's illustrated here with a training set in which we have NIRS, and we analyze sample by a biochemical wet lab approach. We calibrate a model and NIRS equation with partial least square or other approach. And by this way, we obtain protein contents and so on. And after, we can use this NIRS equation to predict some new samples without strong hard job to do biochemical analysis. And we can obtain starch content, oil content, and water protein, and so on. And what is good news for that is that there is development of automated high throughput port-averted device for NIRS, but also for evaluated seed morphology. And there is 2 here illustration with a robotized device for at module, but there is plenty example. And with that, we can use GWAS to evaluate, to identify some regions involved in oil content, like linoleic oil content. And this was illustrated here on analysis with NIRS of 263 French landraces that have been genotyped with 50K array. And by this way, we obtain some loci that are involved in linoleic gene, and we identify no genes on other genes. It works. And the good news is that we can use a genomic selection approach to evaluate biochemical content of seeds using genomic selection approach. We obtain very high accuracy for predicting different agromorphological traits on some traits related to grain quality. And it's a way to characterize land masses maintained in genome for seed compositions. Okay, my last part, if I have time, but I think, is more about phenomic selection. Is a new way of using NIRS to predict traits of association of accession in gene banks. And it's following the seminal work of Renaud Vincent and Vincent Segura that proposed to use NIRS to predict not biochemical composition but traits in the field. Okay, what is the idea? You have NIRS, and you replace NIRS, you replace genotyping by NIRS. And we use NIRS to estimate a similarity matrix based on NIRS spectra instead of genomic data to calibrate models and predict traits using H-blood approach like J-blood approach. And the good news is spectra are heritable, are under polygenic determinism, and capture a part of genotype by environment interactions. And here is the approach. You have an evaluation of traits on omics in with candidate on training population in a reference environments. You have different environments in which you evaluate your traits. You calibrate a predictive models, and you can predict the traits of candidates in various environments. And in phenomic selection, NIRS spectra are not used to predict clinical composition of sample, but infer genetic covariance between genotypes. And in PES, spectra are considered as the genotyping scale as genotyping. And here is a project that we propose to as proof of concept to use it for gene banks. Typically, we try in my non-diff project to evaluate a set of land masses that represent that are a core set of land masses evaluated in a multi-environmental networks in which we acquire NIRS in different environments to estimate similarity between NIRS. And we have NIRS in gene banks that are evaluated by other devices. We estimate NIRS spectra similarity of grain with NIRS spectra from field trials to try to predict traits of land masses in different environments, is the idea. And the advantage of NIRS is very low cost, it's very easy to implement in GenBank because it's and there is automated device as illustrated here by a device from Agap Institute that have been developed to analyze seeds directly from GenBank. And that's all. Sorry for just a few remarks. Genomic selection can be used to predict traits of land masses from GenBank based only on allelic frequency. Is a good news for land masses in which there is large heterogeneity, and is a way to characterize agronomic traits of land masses in GenBank. And NIR is a low-cost, non-destructive approach that are currently used for predicting biochemical composition. You can use GWAS on that, genomic selection to extend to trends on seed composition to predict in GenBanks the chemical composition of your seeds. And it's good news. And last but not last, you can also use NIRS as phenomic selection with phenomic selection to predict agronomic traits of land masses in GenBanks. And we try to do that in microorganism analysis to try to know if it's possible or not. And that's all. And here is all people that contributed to this work. And it's very important, all people that collected, thanks to all people that connected maintain land masses on line collection from the reference GenBanks. It's very valuable resource for readers as well for research. Thanks a lot.
Thank you, Nicolas. We have less than a minute left. I don't know. Do we have time for questions? No. Okay.
It's supplementary.
I'm Karsten Hartelius. I come from the company Videometer, and I'm here today to give a talk on the automated calculation of seed descriptor values and characterization of seeds. The work I'll be talking about is done together with the SQM community of practice. The sponsored mainly by the CGR gene banks. The SQM COP does a lot of research, various types of research within seed quality. So list here that comprises dormancy breaking, improving the regeneration of post-harvest handling, understanding longevity of seeds, and then also various aspects that can be used to optimize the processes in the gene banks. One of them being multispectral image analysis for phenotyping. I will be talking about today. So here is Videometer's instrument for multispectral imaging. It has 19 wavelengths, and it's a calibrated instrument. So it means when you take a picture of an object somewhere in the world, it doesn't matter where in the world, you will always the images will always be the same, no matter where in the world you are. So standardized according to list. But I'm not here to talk so much about the instrument or trying to sell you an instrument. You can relax. But I will talk about this work for the gene banks. First, a quick introduction to multispectral imaging. Normal RGB images consist of three bands of red, green, and blue that you all know from within the visual range. Multispectral imaging, on the other hand, consists of more wavelengths, and not only in the visual range, but also in the ultraviolet range from around 200 nanometers up to 400 nanometers, and in the near-infrared range above the visual range from around 700 nanometers up to 1,000 nanometers. So, multispectral imaging, the purpose is to be able to characterize the object under study in much more detail. Example of why that is useful. You can see on the left we have a RGB image of a bottle with a glass bottle with a clear liquid. It's very difficult, and we want to be able to detect the liquid level, which is very difficult in that image. However, by using instead the multispectral image, we can use some wavelengths in the near range and combine the wavelengths and get an image where it's much more easy to see where the liquid level is. On the right, we have an example from pet food where we have different types of plastic contaminants in pet food. Both the meat and some of the plastics, they have a reddish color, which by the eye in a visual, within the visual range looks quite similar. But again, when looking at the whole spectrum from ultraviolet up into the near, it's possible to combine these wavelengths and get contrast that makes it much more easy to detect the plastic contaminants. Moving into the seed, the area of seed, which is more interesting for you, we have this example where we have two varieties of seed, durum on the left, common wheat on the right, and we can use the multispectral image to create a transformation that separates these two types of varieties. The technology can also be used to detect damages on the seeds that, for instance, different types of mold and fungus that expresses itself in the phenotype. In this case, we have barley infested with fungus or with fusarium and some gray mold, and we can see that we can detect those two types of damages. So those are two aspects, the seed varieties and damages in the seed, where the multispectral image is very useful. Apart from the multispectral transformations, we also have a lot of other, use a lot of other algorithms for characterizing the seeds, shape, contour, texture, size, all kinds of different algorithms combined with the color makes it possible to characterize seeds in quite a big detail. Now I will talk about the work we have done on rice descriptors for rice seed characterization. So the idea here is to try to imitate the seed descriptors that are given in the manual for rice, as many of you probably know, or at least it's used very much from within the gene banks. Try to be able to automatically calculate those descriptors on seeds, on rice seeds. So, and the idea is to have these features first to locate some landmarks, some key landmarks on the rice seed, and afterwards we will collect color and shape information from relevant places on the seed. So what does it mean? On the left we have a rice seed. On the right we have the landmarks detected, so includes the sterile lemma base point at the bottom, the apiculus tip point shown as a red cross at the top. We can also locate the lemma keel point, the tip of the horn, of course, and we can detect the lemma and palea region on the seed. So the idea is to sort of do this on all the seeds as a first step. Once we have this in place, the landmarks, it's much easier to detect all the other fun stuff. So, okay, to start with, length and width, once we have the landmarks in place, quite easy, quite simple attributes to create, as you can see by the red, the yellow lines in this picture. Then we have the sterile lemma. The length of the sterile lemma is one of the important descriptors. We can use a multispectral transformation to enhance, separate the sterile lemma from the rest of the seed. Once we've done that, we can then measure both the color and the length of the shorter and longer sterile lemma. The horn color, with the landmarks, we can identify the region, the horn region, and we can estimate some statistics of the color inside that region, shown here, the last image. One important feature also is the lemma and palea color on, so it's important to be able to extract the color in both palea and lemma region without including the nerve that goes up through the seed. So that's also possible. Then we have rice seed pubescence, so it's the amount of hairs on the seed. We are able, with image analysis, we can detect hairs on the seed, as you can see in the third image. And in the fifth image, you can see hairs that have been detected on the seeds. Once they have been detected, we can also categorize them into short hairs or long hairs. And we can, furthermore, we can separate them in the lower and upper part, because this is required by the descriptor. So that was rice seed pubescence. Then we have also an interesting descriptor here. It's the measurement of anthocyanin level, different parts of the seeds. There's one descriptor that requires that you measure it on the lemma keel. Another one is below apiculus, as you see here, clearly anthocyanin level. So, but with the landmarks in place, you can see it's quite easy to extract the color information exactly where we need it from, in this case also. Now, so here we have a complete list of the descriptors that we have made for rice so far. And so with these features in place, we can, for an unknown accession of rice seeds, we can calculate all these features, and they can be output for further processing. We can also, some of these seed descriptors, they are actually categorical variables. For instance, the color of the seed. There is a descriptor that separates the color, or way that includes 10 or 11 different color categories. Based on the blood features you saw before, we can do a classification, set up a classification method together with the features using machine learning, and we can estimate quite precisely the color categories. So we can both get nominal values, categorical values, and nominal values. Okay, so later the next step in this work will be to be able to feed a sample, an unknown accession of seeds into the instrument. The instrument is scanned and analyzed and compared with a database of different accessions, and then via some kind of similarity measure, we can show the accessions that are closest to the accession that we have just scanned. Hopefully, this can facilitate and be used in the gene banks to improve the recognition of accessions and the analysis of the different accessions. So that's basically it. I just want to say also that we are doing work also. This work was on rice. We're also doing this work now on wheat, cowpea, and chickpeas, as well as maize will come later this year. So the aim is to be able to imitate the seed descriptors for all these crops. And and I have said that the first goal was to imitate the seed descriptors as they are described in the manuals. Ultimately, we want to actually go further, because it's not a matter of imitating existing seed descriptors only. The overall goal, or the ultimate goal, will be to come up with features that can separate as many accessions as possible, going beyond the existing seed descriptors. But it's important to use the existing descriptors as a base, because it gives us a lot of knowledge about where the differences in the accessions are. But we will take it further to so in order to be able to separate even more accessions in the future.
Thank you.
Good morning, ladies and gentlemen. I'm Viktor Sydoruk from Forschungszentrum Jülich. Actually, we were able to develop a quite unique platform which calls PhenoSeeder, which allows to optimize phenotyping and sowing of individual seeds of different sizes. Here you can see as an example of seeds, so some of the seeds that we already managed to use. So here I mentioned several points where the seed phenotyping can be very useful. And one of the critical points is to optimize the phenotyping outlines. We can pre-select, for example, we can sort the seeds or the starting materials before the further use. And we can characterize, for example, the harvest seeds. It can be useful also for seed physiology or for characterizing germplasm. Besides the phenotypic traits, later genetic traits can be determined after selecting the seeds. In this case, it might be also interesting for industry. The PhenoSeeder is a part of our seed-to-plant tracking platform process, and it deals with analysis of seed traits and with sowing of the seeds. Usually they are sown afterwards, after the analysis into the well plates with soil, and later these well plates are moved further to the plant phenotyping platforms, where there is a germination detection platform exists, and then pre-cow platform where the soil, the small seedlings with the soil transferred to the big pods, and then these big pods are transferred to the different different environments, such as greenhouse environmental conditions or climate changes. After the growing, in many cases harvest is done, and the seeds from the harvest can be further used in the cycle. as a second generation, and then the third generation, and so on. All the data, all the IDs, and they are tracked, and actually they are stored in database. What is PhenoSeeder? We try to make our best to deal with everything which is critical for the seed phenotype. And the PhenoSeeder is located in climate-controlled environment. It's based on the cucurobactic arm, and this cucurobactic arm can reach different locations, which is in the closet cabinet, and we are calling these locations as stations. And here, for example, the 2D imaging station where the seeds are picked up, they are basically found on the surfaces, and then they're picked up. Then these seeds are brought to other stations, like for example 3D imaging station for volume carving. There is also an acoustic volumeter to determine the volume, and of course weighing stations for different seed sizes. And at the end, there is a seed placement station where basically the seeds can be sown or sorted into batches. Additionally to the PhenoSeeder, we wanted to include as a station to the PhenoSeeder the seed city, but currently it's a separate station. It's a separate device. Let's start from the 3D imaging station. So the seed is brought to the camera, and the camera is taking photos, which is visible in the middle image. And from these photos, we are able to reconstruct the 3D this 3D model of the seed. We are developing all the time. So the PhenoSeeder was initialized quite some time ago, but we are developing it all the time, and therefore we introduced also the new volume carving. In this case, we were able to increase the speed of operating and plus the performance of the reconstructed models of the seeds. Here you can see, for example, back from 2016, and nowadays data obtained from the herb seeds, where we can clearly see the better correlation between volume of the seed and the weight of the seeds. Here, the examples of seed traits that we can deliver and the reconstructed seeds as a comparison. So the 3D model, together with the 3D models of the seeds, we can deliver envelope volume, convex hull volumes, surface area, length, width, height, sphericity, luminosity, and we're currently working also on the color information, which is also not so easy because you need to determine the lighting and other things. And of course, we are checking the reconstruction quality. Our current actually aim is to develop also the possibility to determine the density quite precisely, and to do that we need to do a proper volume reconstruction and decrease the errors as much as possible. Another station in the PhenoSeeder platform is the acoustic volumeter. It allows to measure the skeletal volume, which is a little bit different from those volume which is obtained from volume carving. Why? Because volume carving basically takes into account, it cannot take all the surface information into account, and the acoustic volumeter allows to penetrate inside the seed somehow. So it measures basically those parts where the air can penetrate. And you can see clear difference, for example, on the two images here, for barley seeds and for bean seeds. For bean seeds, for example, the volumes are quite similar, and for barley seeds, the volumes are different. And using both of these volumes, you can basically say something about surface. After the seeds were phenotyped, actually we have different ways how the seeds can be processed. One way is to recollect the seeds, the specific number of seeds in the batch. And another one is to sort the seeds into batches according to different seed traits. We can also store seeds for further analysis or for further planting or sowing. Here's an example of storing current seeds done by our current PhD student. And we can also do direct sowing, as I said before. During the time of PhenoSeeder operation, we actually, through this PhenoSeeder, 500,000 seeds already, individual seeds already were passed. It doesn't mean that all of them were fully phenotyped. Some of them were just sown, some of them were only weight was obtained or photos were done, but actually we were dealing with many genotypes and many varieties of the seeds. As an additional tool, we have SeedCT. It's this tool actually allows us to phenotype also the internal structures of the seeds. And here we can also go from individual seeds and to batches of the seeds. And here just the nice examples of nice videos of what basically SeedCT can deliver. Get knowledge. Here is my enabling technology group at Forschungszentrum Jülich, and all the people which were involved in PhenoSeeder development. And also I would like to acknowledge our institute. Thanks a lot for your attention.
So good morning to everybody. Thank you for inviting me to speak about our work on seed phenotyping. I'm from Emphasis, from the Italian node of Pinheiro. And we used to work on plant phenotyping, mainly using volatiles detection, VOC detection. So VOC, or better, biogenic VOC, are mainly produced by all the living organisms, plants mainly, fungi, bacteria, humans, of course. They are released in the atmosphere through different biological processes. What they do, the VOCs are different. They belong to different groups. One of the more studied is the isoprene, but there are also monoterpenes, sp-terpenes that are all produced by plants, but not only. They have very important ecological roles, and so first of all, they attract pollinators. So plants use VOC for attract pollinators. That's important for the reproduction. They are involved in defense mechanism, and now we are studying more and more this kind of interaction. And they serve also for plant-plant communication, in general for plant environment communication. I'm presenting here the infrastructure of Emphasis that we have in INRAE, where we used to study VOC detection with this system. This is the PTR proton transfer reaction time of flat mass spectrometer. That is a kind of nose, okay? That is a very high specialized nose. It can detect hundreds of volatiles in the same time, and is highly sensitive. We can obtain perfect trillions of volatiles in volume. And it's useful because it is a non-destructive method of analysis. So here are some of the images of our infrastructure. And in the case of seeds, we are starting right now using these markers for the characterization of seeds. We use that mainly for the characterization of plants. In the photo, you can see in the bottle with the blue cap, some seeds that have been phenotyped for VOC production. Here again, we have this system to checking from plant communication with exchanging volatile from a box to the other, also with some specific work to check if there is a priming effect. Again, okay. So here are some of our results. We use a VOC characterization, of Volatilome characterization, trying to check the difference not only among different stresses, because we used to work with the stress biotic and abiotic stress, but we can characterize, we have seen that we can characterize also difference between different species or different lines or different cultivar. Here I'm presenting results of beans, so the two different lines. S is the susceptible one to Coledotrichum, the anthracnose of beans. The arrow one is the resistant one. Okay, you can see with the S minus and R minus are the control, so the one that are not in interaction with the pathogen. And we have very different volatilome of the two, so we can characterize them. In the meantime, you can see the resistant one have present no difference when is in interaction with the pathogen when there is disease, while the susceptible one make different volatiles, not in terms of quality, but much more in terms of quantity. So this is one of the activities that we can perform, and we also correlate these results with the proteomic and transcriptomics analysis as well. Here is another kind of diagram that we can show. You can say R minus with S minus, they have a different profile of volatiles emitted in terms of, mainly in terms of quantity. So what about seeds? We are just starting with seeds. We think seeds are plants because they are the starting of plants, can be differentiated in terms of volatiles. Can we have volatiles, VOCs, as a markers for adjacent characterization? Here we perform an experiment where we have a different adjacent of pea, grass pea, soybean, and fenugreek, where the control are the seeds with no treatment. The AA is an artificial aging. We put the seeds at 45-60 degrees for 8 hours, and the hydropriming has been in water. So you can see we can easily differentiate the different treatment of the different adjacent. So something happens when you make the artificial aging in terms of volatile emissions, and also something happens in terms of hydropriming. So the seeds change their volatile emission. The idea is also to try to characterize the difference in terms of adjacent, if you can distinguish different lines of the same cultivar. But well, we don't know if it is possible or not yet. Here we have the first result that don't give us in two very good results. But it is known that some of the volatiles that are emitted by seeds are still characterized. Here, for example, they produce ethanol, acetic acid, also acetaldehyde. And for example, acetaldehyde, ethanol, and methanol have been described as a major constituent of the airspace of the seeds when analyzed. So I finish very fast because this is a very new topic, a very new idea to how characterization of plants, of course, but seeds in particular, because we imagine that we can give a new pieces in the bigger puzzle of seed characterization, because all the seeds, all the cultivar, all the different species, they smell different. Probably they can give us markers for their characterization, at least for the characterization of the quality of the seeds. So what we think is that our work can be important in agriculture, so understanding what profile can also aid breeders for the program. Okay, we need to have a lot of data to work on. In ecology, pharmacology, why not? Some seeds have potential medicinal properties, also in terms of volatile emission and food industry. At the moment, we are waiting. We are emphasis. We are in the agro-service project as well, and we are waiting for the result of the Biota project. This is in common with Carla for seed characterization with volatile. So we hope that the project will be financed, and for that we are in agro-service. The new call is open. If someone would like to apply with us, we are here. Thank you. Thank you.
Okay, thank you to all speakers this morning. We will have now some time for questions and answers. Do you want to start from the room or from online? You have Francois here.
I actually have a question to Michelina. So I understand that the volatilome depends on the genotype, but also the condition in which the plant was grown, and also probably of the condition perhaps the environment as well. So how can we deal with all of that together?
It's not easy, of course. I imagine that we need to have many data recorded of the different environment, different conditions. For example, if the seeds are disease or with a pathogen or not, if there is a virus, for example, maybe this can be a very fast and not expensive way to check. But we have to, we just started. So we need that can be further, many.
But it's
I think it's
it's really well.
the best. Thank you.
We have Giovanni here.
Very nice talk. I have a question for Karsten. How many seeds do you need for Karsten Hartelius? How many seeds do you need for phenotyping? I saw you use petri dishes, but can you do it on a smaller number?
So the question is, how many seeds Karsten needs to do phenotyping?
It depends on. It's a question we always get, and it's difficult to answer in general because it's difficult to separate the. But typically we are using with Africa rice. As you saw a presentation from Africa rice. 50 seeds of each variety.
30.
50. 1, 5.
One question for Michelina.
Yes.
So you assume that seeds are not very active metabolically.
That's not true.
You assume. So I wonder if you have done a time course of volatile emission over the time. And have you tried any inhibitors of metabolic activity? Does any of them between emission, things like that.
Okay, actually, here I'm presenting results of 3-4 weeks ago. So it's really in the beginning. But it's our idea to have a time course of the also germination line, and then also going to the at plant level. So checking what's happening. But yeah, seeds it is assumed that they are not so metabolic active, but that's not so true. Some of them you can smell them. And the smell comes from something inside the seed. So everything is starting. We hope that we can really help. Also, data bank, because if we will be able to, for example, find markers for aging, for example, we can think to put sensor for those markers into the gene banks and say, okay, in that part of the seeds of the gene bank, those seeds are in aging. So take care. It's something like that.
So we have Anna Maria there.
There's also a couple of questions from online, so we will also get to those in a second.
It's for Karsten. We just bought a Videometer. We are using it. We are training commonly seeds. Can you use the information you have for the other species already? Because we are gene banks, so we have all kinds of species conserved. Can you use the information you have already trained for those species?
The work I've presented wasn't right, so the seed descriptions for rice are more or less in case to be used by other gene banks as well. With every rice, but they are usually they are testing. They're doing testing also now, and to be ready to have others to use them also, which we very much welcome.
Okay, so maybe I will go to the online question. So there's one from Charlotte Allender, and to Karsten and Viktor, what is there any further work underway on seed imaging and physiology concerning dormancy viability? And I think that maybe follows the question to Michelina.
Thanks a lot. So it's any further work for the seed physiology? Yeah, actually we have different partners, and they're basically doing seed phenotyping using our platforms, and then they're using those data to connect it to the physiology. But we are personally, for example, we have work, the safety work, which is a PhD student who is trying to link the, for example, the heat treatment of Rhabdopsis seeds, Rhabdopsis plants and treating of plants with further genetic analysis to link those treatments over generations. Me personally, I'm not doing that because I'm more into the development side of the PhD. I'm a physicist, and actually I have another tasks. But actually we have partners who do that.
Yeah. Right now we don't get reach, but there are a number of articles and that can be downloaded that show some of the results on dormancy and predicting germination. I can provide some links afterwards on the chat if that's relevant. dormancy and germination.
Yeah. I think that would be very good, and we can probably provide them through the website. We are running late.
Yes, but we started at 11:10, so.
Maybe one question, Luis?
Related to germination, I was wondering if you had we were in Braga looking at the Videometer A. We were discussing about how you can predict germination, and you can predict, can use machine learning for fresh seed, but what happens with those that are already in the chambers? And if you have talked about other characterization ways using hyperspectral analysis, have you see any difference in germination with this technique? We see the seeds after aging that they become brownish. Is possible to see something?
I want to say it's difficult to take a seed, a dry seed, and predict whether they have to mean it's image analysis. It's not magic. It's like when a person is born, you cannot predict whether he will be a pilot or whether he will be a farmer. No, it's the germination properties. They have to express themselves somehow in the phenotype. So that's what we have to do with these technologies. We cannot see inside the seed. We can see what happens inside the seed, but it has to be through some expressions on the outside. So it can be on the size, and shape, and the discoloration, as you mentioned yourself, of the seed.
Sorry, I just want to add, actually germination also depends on the storage conditions of the seeds, and this is known. And actually, we are also currently developing the station which allows us to measure the moisture of the seeds. This is moisture is quite important in this case. And if the seeds are getting brownish, we got an impression of these Arabidopsis seeds that is a very deep tree, because in case of Arabidopsis seeds, we got that the seeds were getting dark when they were asleep. I mean, during the development process of the plants. So maybe it's related to the environmental conditions which the plants.
I think for further questions, you will also be able to contact the speakers directly.
So I would like to make one final question to Nicola. I'm taking my chair privilege. So Nicola, you showed us about this maize program. So could you please share with us what do you think are the benefits of using phenomics in this process?
The advantage. Maybe I can speak. Okay. Yes, the advantage of using phenomics is just that if you imagine that you have plenty of seeds in gene banks, you can use NIRS, or you can predict all the traits that you want by using a training set in the field for different traits. And this is the advantage of phenomics. The cost is close to nothing because NIRS is like €1. Genotyping is like 20 or 50 euros. On the main cost is field trials. On phenotyping in field trials. And if you can extend for the characterization of non-RAS maintained in gene banks for just by using phenomics approach, your cost actually is very close to nothing. On the other advantage is that a lot of people have NIRS in gene banks for different reasons.
on a lot of breeding company have this for breeding program implemented in breeding programs. On the accuracy is not so bad.
Okay, thank you. So let's welcome our speakers again. Thank you so much.